Today's lab's going to be one of the ones we're a little bit different. I am going to kind of lecture for a little bit, then we'll start something. Then maybe I'll lecture for a little bit more, and then while there's going to be an incubation period and some other stuff going on. And then I'll lecture a little bit more. There'll be another incubation period. I might lecture a little bit more or just talk about the next part real briefly. But also there'll be some downtime that's set aside for you guys to do your colony counts for your plates, your group projects that you did last week then. Today's lab is what I kind of call DNA fingerprinting. And I do mean fingerprinting, just like your fingerprints. Does everybody have the same fingerprints, or is everybody's fingerprints considered kind of unique? Unique. And so we are going to be doing a little bit of a lab with DNA fingerprinting, almost like it's a crime scene, for instance, then as well. So today's lab, again, we're going to focus on data collection from last week's group projects. So you do your colony counts. Those are going to be used on the UV yeast paper as well, which I will probably talk a little bit more about here towards the end of class today, but also that's posted to Brightspace. A description of that is posted to Brightspace as well as the Dropbox for that then. We're also going to be doing this DNA fingerprinting lab. This is going to be kind of an example of a crime scene investigation, so CSI. What I'm going to give you guys is, in essence, a mock crime scene DNA sample. And then also you're going to get, actually, what do I have 4 up here, but you're going to get 5 DNA from 5 different suspects, suspect 1, 2, 3, 4, and 5. And what we're going to do is a little process by which you can kind of see whose DNA matches up with the DNA taken from the crime scene. Which one of the suspects has DNA that was at the crime scene, or in essence is kind of the culprit potentially of a crime, for instance. But before we get started, what crime do you think we should be investigating? Right.
Whoa.
Hunter went there. Okay. It would potentially be used for that. Let's not do that one.
A murder.
Yeah, we can do a murder. That's sadly enough, that's better. All right. So been doing this for like 8 years. First time. All right. All right, so what we might have here is maybe we have a DNA sample from a crime scene. Again, maybe we have then collected DNA samples from 5 different suspects. What we want to try to figure out is which one of the suspects' DNA might match the DNA that's also been left at the crime scene, for instance. With that, though, does that necessarily prove that the suspect, say if it was a murder, that they committed the crime?
No, someone could have taken their DNA right off of them and dropped it.
So you could have somebody maybe being framed, I suppose, yeah.
Somebody could have been there.
Yeah, really, it kind of depends upon the context. Like if you're finding the suspect's blood, for instance, at the crime scene, there's a bit of a chance that, yeah, maybe they were involved in like a fight or something like that, for instance. But it really depends upon the context. Usually when DNA is found at a crime scene, it just places the suspect at that crime scene at some point. But it depends on the context of how the DNA was found. Was it a blood sample? Or was it just like a hair sample? Again, the hair sample just means they were probably there at some point, for instance. So it doesn't necessarily mean they did the crimes, but it's still useful as a data point when investigating these types of stuff. DNA. We actually talked a little bit about DNA last lecture. But if we look at DNA in a couple different ways, again, it's a polymer of nucleotides. It's double-stranded. A single-stranded DNA is held together by covalent bonds, what are called phosphodiester bonds, hold together a sugar phosphate, sugar phosphate, sugar phosphate backbone of the DNA. But DNA is double-stranded, but it's asymmetrically double-stranded. It's an anti-parallel double helix. So there's 2 strands of DNA, but one runs 5' at the top to 3' at the end of the bottom. The other one runs anti-parallel to that, where it has the 3' end of the top and the 5' end of the bottom. So they run in opposite or opposing anti-directions then. And then what kind of bond holds together the two strands of DNA? Hydrogen bonds. And specifically, A pairs with T, an adenine on one DNA strand pairs with a thymine on the other one, and G pairs with C. So a guanine on one strand will always pair up with a cytosine on the opposite strand then. So that's the base pairing. A pairs with T and vice versa, G pairs with C. Okay. Now, with that though, is there any real variation in the sugar phosphate backbone? It's phosphate, sugar, phosphate, sugar, phosphate, sugar, right? Same thing over here. So what is different though? How can you detect differences in a DNA? Say the DNA of one suspect versus another suspect versus a third, fourth, fifth suspect. If it's not the sugar phosphate backbone, where can you see some variation or some differences then?
The codons.
The codons, which are made out of what? Nucleotides, and specifically nucleotide sequences, the orders of the A, T, C's, and G's. So what part of the DNA can you compare? It has some similarities, but also contrast, has some differences. Sometimes the ordering of that C's, T's, G's, the ordering of the nitrogenous bases can differ from one person to another, to another, to another. Don't get me wrong, your DNA is probably 99.9% similar to everybody else sitting in this class. But there are going to be some, there's still mean there's some differences there, and they're detectable differences via some different kinds of techniques. But that also means everybody's DNA sequences are unique. Remember, DNA is information. DNA is information that codes how to put your cells together, but also how to put you together. And if you don't look exactly like somebody else, that means you have differences in your DNA, differences in that information. And we could detect those differences via some other techniques. So that, oh, but the variable part that we need to look at is the order, the sequence of the A, T, C's, and G's, the nitrogenous bases within the DNA. Then. Questions about that? Now, how do we do this? There is different ways to detect DNA sequence differences. One of them is, well, maybe we just look at a whole genome comparison. So nowadays we have the technology to, for instance, take a human genome and sequence all the A, T, C's, and G's and the order that they are in your particular body, in your cells. And we could do that. And that would be the most accurate. Get all the DNA sequences and the order of those DNA sequences and compare them one versus another versus another. The problem with that is it's pretty expensive and time consuming. So for instance, the very first time this was done, does anybody know what year the very first human genome, all the DNA sequences of one particular person was sequenced for the very first time?
2010.
Little bit further back than that, but not too far off.
2003.
Yeah.
Huh?
Huh? 2003. Took about a decade. Took about 10 years of work by scientists. Cost a few $100 million to $1 billion. But that was because to do that, they had to basically invent new technologies that did not exist previously. And that project was heavily funded, heavily worked on for years. And it turns out most of the time consuming and expensive work was at the front end of the project. By the end of the 10 years, they were humming along because they had actually built some new ways to look at the DNA of organisms and to sequence and look at that DNA of organisms in exacting detail. And that technology has only increased. Nowadays, I said first human genome was about $1 billion, 10 years. Extremely expensive and time consuming. Now they can sequence a person's genome in about a day for about $3,000 to $5,000. That being said, do police departments and crime scene investigators have usually $3,000 to $5,000 to throw at one single crime?
No.
No. So that's not what they do. It's not real. It's basically not to the point where it is cheap enough where you can do whole genome sequence analysis on, say, 5 different suspects and drop $15,000 on that one, for instance. Does that make sense, everybody? So we have to do some faster, less expensive techniques. And it turns out it's not necessary to sequence entire human genomes. That's a level of detail we don't need to go to determine what is called a DNA fingerprint. Compare the DNA from one person to another to another. We can instead do a different process, and you guys are going to mimic doing that today. So we're going to do this one. Instead, the DNA from a person is isolated and treated with a restriction enzyme. And I'll talk more about that what in a second that is, but in essence, a restriction enzyme is an enzyme that cuts DNA. And I'll fill in some more details on that. And what the restriction, well, no, I'll go through it right now. So you start off, you isolate DNA, and then you could just do this with a hair sample or like swabbing the cheek cells. If you constantly, your cheek cells are constantly what's called sloughing off. So you just rub a swab against it, you get tons of cheek cells with tons of DNA in there. For instance. Now, what you do is you isolate that DNA from a person, and then you treat that DNA with a restriction enzyme. And a restriction enzyme is some type of protein enzyme that cuts the sugar phosphate backbone of a DNA molecule. But it doesn't cut the DNA everywhere or anywhere. It cuts it at very, very specific spots. So for instance, one type of restriction enzyme, and one of the ones you guys are going to be using today is EcoR1. By the way, that just stands for E. coli restriction enzyme number 1. It's a specific restriction enzyme that's from E. coli bacterial cells. But we've isolated it, purified it, and use it. It binds to the sequence of GAATTC. If you guys look in here, there it is, GAATTC. There's my little sequence in my DNA molecule here. And again, my DNA is double-stranded. So here's one of the strands held together with the other strand. So that restriction enzyme, that EcoR1, will only bind to DNA at that specific sequence. So here's my EcoR1, it comes onto there. It binds to that GAATTC sequence. Once it DNA sequence, it activates it and it causes it to cut that DNA sequence. In this case, it also does an uneven cut. It cuts the top strand here, but it leaves a little overhang, and over here, again, it leaves a little overhang because it cuts the 2 strands asymmetrically as well. So it cuts right after this G, and then also right before this G, right before this A over here. But what it does is it cuts both of the strands of the double-stranded DNA molecule. Questions about that? By the way, why would E. coli have an enzyme? This enzyme is something that E. coli bacterial cells have. We found them in the bacteria, and now we use them for some other purposes. But why would E. coli have an enzyme that cuts DNA?
So like penetrate like your immune system?
That is a good guess, but it's not the right answer. That is actually a good guess, but it's not going to be what's going to happen. E. coli is actually not an intracellular predator of your cells. It doesn't get inside of the host cells. It stays on the outside and kind of destroys them from the outside. So this is one that I don't necessarily expect you. This is actually a simple version of a bacterial immune system. This restriction enzyme from E. coli helps to protect the E. coli cell from its pathogens, and specifically viral pathogens. So what happens is if a virus, what happens during a viral infection of a bacterial cell, the virus docks with the bacterial cell, but then injects its DNA into the E. coli bacterial cell, and then it hijacks it, the bacterial cell, and makes it into a virus factory. And that's kind of what viruses do. They get inside a host and they cause the host cell to start making viral copies instead of the copies of the host cells that it normally does. What happens here is when it injects its DNA, it's injecting double-stranded DNA into E. coli. And guess what? It's E. coli restriction enzyme 1 digests the viral DNA and destroys it in an effort to protect the bacterial cell. So it's a very simple rudimentary immune system for bacteria. That is what it's involved in. Yeah.
Why does it cut like that? So like the top piece, I understand at the bottom piece, why does it?
Short answer, it just does. There are actually some restriction enzymes that leave these overhangs. There's some other types of restriction enzymes that leave what are called blunt ends, where they cut the strand at this bottom and the top strand at the exact same spot. It just has to do with the nature of the restriction enzyme itself. There's no particular reason for it. Yeah, Jacob.
So does that mean E. coli doesn't have that sequence in its DNA?
Great question. It does. But its sequence is often methylated. It adds a little methyl group onto the C's. And that means, and the viral DNA is often unmethylated at the cytosines. And what that does is it protects the methylated DNA in the bacteria, has a little methyl group that's added onto these after DNA replication, and that protects them from the enzyme. So this specifically also targets not only the sequence, but also unmethylated DNA, which is usually then also an indicator of viral DNA. So that's actually a great question. So it's a little bit more complicated. Okay, now where do restriction enzymes come into play with this whole DNA fingerprinting lab? Well, what you can do is, again, you can isolate DNA from suspects and from a crime scene sample. You get all the DNA from a person, but then you treat it with restriction enzymes. And say, if you treat it with EcoR1, anywhere where there's that GAATTC sequence, it cuts it. And what you do is there's a bunch of these sequences in your genome everywhere, literally. They're fairly common. And so what you get is a ton of different DNA fragments produced. Remember my cut before, I just had one sequence, so 2 fragments produced. 2 double-stranded fragments were produced. But now just imagine you have tons of these fragments that are produced. And these are a bunch of fragments of different sizes that were produced. But you treat them, you get these differences in the DNA sequences. It could be that somebody has a mutation that introduces a new E. coli 1 R1 binding site over here. This other person doesn't have that binding site. They get different fragment links. It's actually fairly common for people to have what are called chromosomal inversions. Most of the time, your DNA sequences overall are the same, but sometimes part of somebody's chromosome decides it just wants to flip around compared to somebody else, and you get different size fragments produced. And so, for instance, you might have like person A versus person B versus person C versus person D, and you get different, you see banding patterns, different fragment links, but they get their own unique little fingerprint or map that produced of their fragments. So it generates DNA fragments with differing links that we can visualize. We separate those DNA fragments based upon the size or the length of those fragments, and we generate what is called an RFLP map. An RFLP, because everything's got to have an acronym, is restriction, fragment, length, polymorphisms. And again, this is very descriptive. Fragments generated by restriction enzymes, their length, and polymorphism just means many shapes, in essence. Many different patterns is what that refers to. So you can see these different patterns, these different banding fragment lengths, and you generate one of these maps. And that can tell you one person versus another. It's different enough that you can even tell siblings versus parents relative to one another as well. Okay, not identical twins, though. Identical twins are called identical twins because they have the exact same DNA, for instance. Questions about that? Okay, so what are we going to be doing today? We're going to be starting off with a few of the things. We're going to be using some mock DNA samples. These are shortened versions of an individual's genome. I'm not going to actually have you isolate DNA from like yourselves or other people. I've got some easier fragments to work with, make it a little faster, a little easier to do. So these are shortened mock versions of an individual's genome. We're going to digest them here in a second. And then we're gonna digest them with those restriction enzymes. And then we're gonna sort those different possible DNA fragments based upon size, doing something called a gel electrophoresis, which I'll talk more about here in a second. And then we'll visualize that. So I said 4 large groups, but get into 2 groups. Since our attendance is always, I don't know why I always say 4 groups. I never get 4 groups the day before Thanksgiving break. But get into 2 groups, 3 and 3, it looks like. Then you're going to turn to page 77 in your lab manual and start the protocol for doing a restriction digest. Do I have 77? I think it's actually, no, it's 77. So it's top of page 77. It says protocol for restriction enzyme digestion. You're going to start doing this. I have the samples up for here for you guys to work with and follow it along. We're going to get that set up. But once you mix together your tubes, they have to incubate in the 37 water bath over here for 30 minutes. It takes about 30 minutes for your restriction enzymes to cut the DNA fully. Does that make sense? And they got to be at the right temperature while they do that. So go ahead and get into 3 groups to get that set up. One thing I want to make sure though, be very, very, very careful when pipetting today. You are pipetting much smaller volumes, like 10, 12 microliters than you guys have pipetted in the past. What that means is you're going to press down on your plungers, your pipetters, and it's barely going to move. And that is the way it's supposed to be. It's press and you're already there, basically. Does that make sense, everybody? If you're not sure, show me. You're pipetting very tiny bits of liquid this time around. And a lot of the earlier lab sections were just like, and then they ran out of their enzymes and stuff like that. So it's very tiny, very small presses is what's going to be involved here. Questions? All right, go ahead and get going. Okay, now that we got the enzyme mixes going, then we're going to talk about the next part. So again, what we're doing right now is we are digesting the DNA. So we're taking those restriction enzymes and we're creating those restriction enzymes are cutting the DNA into fragments. But again, you get fragments of different lengths. So you can kind of see that difference where you have different DNA fragments being produced of different lengths, where you can see these restriction fragment length polymorphism maps. So right now, we just set up our DNA, mix it with our restriction enzymes, and we're generating those DNA fragments of differing lengths. The question is, now if we have these DNA fragments, how do we actually separate them apart? And how do we actually visualize DNA as well? Can you guys see DNA?
No.
Nope, it's tiny, it's microscopic, it's too small to be seen easily. So we need a technique that lets us actually see the DNA in some way, but also will separate out these DNA fragments based upon the size or the length of the DNA fragments. So we can even generate a map like this in the first place. So how do we separate DNA fragments? By size, by length. This is a process called gel electrophoresis. I'm going to talk a little bit about its purpose, how it works, and then how do you actually visualize the DNA to, in essence, see the DNA then as well. So what is gel electrophoresis? What is the purpose? The purpose of gel electrophoresis is it's a technique, a biological technique, a biotechnology that is used to separate nucleic acids or sometimes also proteins based upon the size of the DNA molecule, the RNA molecule, or the protein molecule. And what it does is it uses a gel, and this is a little bit of a gel right here, but I also have some gels that you guys are going to work with here in a second. It uses a gel. This is why it's called gel electrophoresis. And it literally is basically kind of like slightly thicker gel. It's that type of consistency, a little thicker, a little more, less wiggly though. But it's a technique, it uses this gel, you load your, say, DNA onto wells in the gel. I'm going to turn the lights down because it's kind of hard to see otherwise. Once. You guys see these wells right here? Yeah. These are literally little wells. The bottom's still sealed, so it doesn't go all the way through the gel, but it's little indentations or wells in the gel. You load your sample into the well. And then what you do is you apply an electrical current to the gel, and that electrical current is going to push / also pull either the proteins, the DNA, or the RNA, whatever you loaded onto the gel, and it pushes it through the gel matrix, through the gel itself. But because of the way the DNA interacts with the gel, the DNA molecules also get sorted into bands or fragments based upon the size of the DNA molecule. So I'm going to talk a little bit quickly about how this works. First off, we're going to focus on this electrophoresis, just to say DNA or RNA, nucleic acids, because those are pretty similar. I'll kind of ignore the protein part, but you can do a modified version of a gel electrophoresis for separating protein molecules as well. Again, we're going to use an electrical current, though, to push/pull the DNA through this. Why can we use electrical currents to move DNA or RNA through a gel? There's something about DNA and RNA that will make it interact with electrical currents and electrical fields. Why would DNA interact with an electrical field if we're looking at the structure of DNA? It does have the hydrogen bonds, but in this case, the hydrogen bonds are a fairly weak bond, doesn't have a lot of charge separation, for instance, but they're actually, the hydrogen bonds almost kind of, it's a partial positive on a hydrogen, a partial positive on a hydrogen interacting with a partial negative someplace else. So those kind of balance one another out. But there is something that is charged about DNA. It's not the oxygen, actually, although that's kind of part of the component. Phosphates. What do you see as the electrical charge on the phosphates? Negative. Here's a negative phosphate. But remember, there's a sugar phosphate backbone. So phosphate, sugar, phosphate, sugar, phosphate, sugar. That's the backbone of a strand of DNA, and every phosphate carries a negative charge. And guess what? Sometimes DNA is thousands to even millions of nucleotides long, and each phosphate is negative. DNA is actually RNA, but as well, very negatively charged because of the sugar phosphate backbones. But charged things will interact with electrical fields. And what we do, in essence, is we apply an electrical field, and we're going to use some power supplies. So I have one over here and then one over there behind Hunter, for instance. Those are power supplies. But what we do is we hook them up. There's little electrical cords, but we put a negative charge at the top of the gel, and we put a positive charge at the bottom of the gel. So we have a cathode at the top, negative, anode at the bottom, positive. But DNA is negatively charged, as I said. So how does a negatively charged DNA interact with the negatively charged field here at the top? What ends up happening? Negative charges do what to one another? Oh, they repel one another. So by putting a negative charge over here at the top of the gel, it pushes the DNA down away from there. And it's also simultaneously getting attracted to the positive charge, because unlike electrical charges are attractive to one another. Does that make sense, everybody? So if we put a negative charge at the bottom, that pushes the DNA through the gel. We put a positive charge at the bottom, negative charge at the top, that pushes the DNA through the gel. We put a positive charge at the bottom, and that pulls the DNA to the gel, which is why I said push-pull forces here at the very beginning. So you apply a consistent electrical field with that power supply, and that forces the DNA to migrate through the gel matrix. Then. Now, DNA with more nucleotides are just longer. They're bigger. DNA with less nucleotides are shorter. They're tiny. The DNA size of the DNA molecule is usually directly related to the length, the number of nucleotides joined together. Do you have 500 nucleotides joined together, or do you have 1,000 nucleotides joined together? DNA just gets a little bit longer, right? Now, what ends up happening though is DNA with more nucleotides are bigger, and therefore they also, because they're bigger, have more trouble moving through the gel. Why would bigger DNA molecules have more trouble moving through a gel? It's not a trick question. It's actually one of the easiest things I'm going to ask you probably this semester.
The bigger it is, the harder it is to move.
And because it's doing what? If we're trying to move through a very thick viscous gel, a bigger DNA molecule gets caught up and hits the particles within the gel. The bigger the DNA molecule, the more it's hitting the gel matrix, or in essence, it's creating friction forces. And as you know, friction is going to slow stuff down, for instance. So the bigger the DNA molecule, basically it's bumping into the gel molecules more often. That generates more friction and it moves slower as a result. The smaller the DNA molecule, it just doesn't hit the gel matrix as often. You get less friction forces slowing it down and you get faster movement as a result. I say my example of this, think of an elephant versus a mouse moving through a thick forest of trees. If you have a mouse, it's got no problem moving right between the trees and its path is going to be pretty straight. It moves pretty quickly. You got an elephant moving through a forest of trees, it may only have to take some pretty meandering paths to force its way between some trees. Might even have to tear some trees apart or anything like that, uproot them. But that really slows down the elephant's movement as a result. And it's the same thing here with the gel electrophoresis stuff. So what you're looking at the end of this is, here's the wells that you first loaded. This is your top of your gel, is the way you can think of it as. But if DNA fragments haven't moved as far, that means they're bigger. So longer molecules are closer to where you loaded them onto the gel to begin with. Smaller, shorter molecules migrate a lot greater distance over time. So shorter molecules are going to be towards then the bottom of the gel, away from the wells where you loaded your sample.
So the farther it moves down,
The shorter it
does
is.
it get shorter as it keeps going? Like-
No, the fragment length is set,
Okay,
but short fragments can move faster. And therefore they're going to be at the bottom. But no, the DNA is not getting torn apart in the process. The only thing that is generating the fragment lengths or tearing apart the DNA is the restriction enzymes that you're doing right now. But we're going to let them digest all the DNA molecules as fully and completely as they possibly can to start with. Remember, they only cut at very specific fragments, sequences. Yeah, so once they've cut all those sequences, they can't cut anymore. All right, so at the end of this, we have DNA in a gel. We've sorted the DNA based upon the size or the fragment length, but we still can't see it. DNA is tiny, and we cut it, so it's even tinier, right? And so we need a way to visualize this. So we actually have to stain the gel in some way in order to visualize it. For DNA and RNA, we often use a compound called ethidium bromide, and it's already in the gels that you guys are going to be working with. So the ethidium bromide stain is preloaded into the gels, in essence. Ethidium bromide is a chemical that binds DNA. So it'll bind to DNA. Great. So, and then also it fluoresces under UV light. So what you're seeing here on the gel is you're not seeing the DNA directly. You're seeing ethidium bromide bound on my DNA fragments. But then we put UV light and it causes it to fluoresce this pinkish purplish color. And pretty brightly actually, but enough that we can see it and visualize it. So we're going to use UV light, a combination of ethidium bromide and UV light to then visualize our DNA and record results then. That being said, you need to be really important when working with gels to have ethidium bromide. Ethidium bromide binds DNA. That includes your DNA if it gets inside of your cells. Again, ethidium bromide is a stain that fluoresces UV light and allows us to visualize the DNA, but it binds to DNA and can damage DNA when it does so. So ethidium bromide is a mutagen. It can cause mutations in your DNA sequences. And if you get the wrong combination of mutations, you can get cancer. So ethidium bromide is both an example of a mutagen, but also a carcinogen, a cancer-causing chemical, for instance. What that means is when we're working with the gels, you're always wearing gloves. Gloves are up here on the front shelf. Gloves are required for today. Once we start working with any of the gel electrophoresis equipment, we assume all that stuff is contaminated with ethidium bromide, and you should always have gloves when you're working with it. Okay. When you're done with gloves, they go into the biohazard waste can, the small one up here at the front. Also, Don't be the person that puts on gloves, then comes and sits down and starts writing in your notebook with your pen, and then puts your pen in your mouth while you're chewing. It's not a strong carcinogen, but we want to just prevent any exposure to it. It's just something you got to be aware of. If you're going to come back and work with something that's not your gel, your gloves have to come off. Okay, and then you're going to have to probably get a new pair when you're ready to work with the gel again. Questions about that? All right. When the gel is done running, we're going to run it. It's going to take about 30 minutes. So we still have about 15 minutes left on our digests over here. Once those are done, we're going to load them onto the gel. The gel's then going to have to run for about 30 to maybe 45 minutes. I'll keep an eye on it. But then at the end of that, we'll be able to take a look at it. And we're going to basically just generate our own miniature version of an RFLP map. What you're going to have is you're going to have a particular fragment pattern for the crime scene DNA. And that particular fragment pattern or fingerprint is going to match one of your 5 suspects. Does that make sense, everybody? And then you know who the possible culprit of our murder is, right, Hunter? Okay. Murder. All right. Couple then quick things before I kind of let you do more colony counting stuff. Assignments for next week. I just want to make sure that you guys know what's coming up. Pre-lab worksheet is due. There is going to be a pre-lab worksheet for the lab. There was actually a pre-lab worksheet due today. So if you didn't do the pre-lab worksheet for today for a pre-lab number 9, please make sure you do that as soon as possible before you forget. Pre-lab worksheet number 10 is going to be due on Tuesday, November 29th. And this is going to be on a lab that we're doing on mitosis, meiosis when we come back from Thanksgiving break. Also coming up fairly quick is the UV yeast paper. We don't have a writing day like the last time. That's because you guys have already written one full paper and have graded those and gotten feedback to everybody. If you have questions about that feedback, please, well, first, absolutely have to look at the feedback. And if you're having any trouble with that whatsoever, please talk to me. I'll help you work through it. If you have any questions about the feedback, please make sure you are emailing me or stopping by my office to get help with that. The feedback is meant to help you do better on the next paper. That being said, you have to incorporate the feedback and you have to work on the next paper. I do have some people that score a lower grade on the second paper if they don't do those processes, for instance. Okay, so please, that feedback is there to help you and to make this second full paper not quite as bad in terms of just your comfort level, the amount of work you need to put in. I just don't want it to be harder than it needs to be, for instance. Okay. The UV yeast paper is due Friday, December 2nd by 5 p.m. So end of the week on that when we come back from Thanksgiving. That does sneak up fast because, well, it's Thanksgiving break starting tomorrow. Need help? Make sure you start early. I will take a look at drafts of individual sections or the paper. We'll offer feedback on those before you have to turn in the final copy. I also did want to talk a little bit about what goes into the paper a little bit as well. So when we look at this, for instance, if you go onto the Brightspace course page, under the writing assignments, there is the UV yeast full paper module, and that has the full paper Dropbox to turn in the paper, but also it has a description of the assignment as well. Here is the class data sets to use on the assignment. This is all posted to Brightspace. Okay, but this is the data set from the percent survival data. I've given you the percent survivals already calculated. I'm giving you the percent mutations already calculated. But that's the 2 of the parts of your paper. The third part is your group project that you did last week and are collecting colony count data for today as well. If you have group members that are not here today, can you please be nice and go ahead and send them a document that has the group colony counts on there? Okay. With this one, I'm not trying to be hidden. Really, there's 3 different parts of the paper. The catalase paper had three different parts: concentration, temperature, pH, right? This one also basically has three different parts and three different figures that are required for the paper. One of the figures should be percent survival versus your time of UV exposure. But you're doing the class data set, not just your individual group data set. So you're using the class as replicates, and you got to find the averages. You're not graphing individual replicates. You're graphing the average of all the different class groups. So you should have one bar that's the average of the 0 second, one that's the average of the 30, average of the 60, 90, 120. But that first figure should have 1, 2, 3, 4, 5 bars in it total. And it should be the average percent survival at 0 seconds versus, and then so on and so forth. Does that make sense, everybody? So if one figure is going to be average percent survivals versus UV exposure times. Second figure is going to be average percent mutations versus UV exposure times. And then the last figure is going to be a figure over your group project data. But again, everybody did replicates for the group project, so it's still going to be averages for that then as well. Any questions about that? But everybody's, and remember also, this is all individual work. The paper, I know you did a group portion together. Your group, once you got the data today, is dissolved. So do not work on the paper together. We've had some plagiarism issues in some of the other sections, for instance, just putting that out there. I'm not saying who or anything like that, but there has been some cases where we had some plagiarism that's gotten caught this year, for instance. But remember, the paper itself is individual work, and everybody needs to turn in their own copy of their own paper. Questions about that? So 3 different figures, 3 different parts, and that's not a bad way to break up your kind of results and discussion section in particular, is to have maybe a paragraph on percent survival, a paragraph on percent mutation, and a paragraph on your group project, which usually involved in some way protecting the cells from UV light, I think, for pretty much all of you, which also means you got a ton of colonies to count today, unfortunately. Except for maybe Jacob and Keegan. Do you have relatively few? Yeah, it is. Okay. So also make sure, again, Friday, December 2nd is when it's due. The rubric also goes through exactly what I'm looking for. I am wanting the abstract to usually be a minimum of 150 words to a max of 250 words, for instance. I am wanting the paper to have correctly cited information, particularly in the introduction section and the discussion section then as well. Should be a number of different things. You should have figure bars. Those figures that you generate should have error bars on them, the same as your second statistics assignment. So there is specifically points built into this paper to have correctly formatted and added error bars to each one of your 3 figures. Okay. Should have citations in the discussion section. And yes, you do still need to have at least 2 primary or secondary references at a minimum as part of this paper, just like you needed for the catalase paper as well. Remember, those are professional journal articles. Your textbook, your lab manual is okay to use as a source, but it doesn't count as one of my 2 required sources. They need to be journal articles, either primary research journal articles or secondary review articles from a professional journal. Questions about any of this? I'll also give you a hint for, since you guys are here, what kind of information probably is the type of stuff you need to talk about in the introduction section? What's the focus of the big concept that you're taking a look at in this paper? Yeast could be part of the intro, but probably not until you're getting pretty specific to your experiment. So yeast could be in there, but that's not going to be the big concept.
So mutation.
Mutation is going to be part of it. Survival is going to be part of it, but survival and mutation as influenced by what? UV light. And there's your big one. How does UV light impact living organisms? That should be what your introduction probably is focusing on. And what that means is, what the heck is UV light? For instance, and how does UV light potentially impact cells? And then also that we're going to do an experiment to explore that and quantify that. So your big focus is going to be on ultraviolet light. What is it? How does it impact living organisms? Questions about that? That's the part that's absolutely critical that you have as part of your introduction section. And usually you could spend even a couple full paragraphs just on ultraviolet light, for instance. Okay. But that also requires you to do a bit of background research that gets worked into that introduction section. Okay. Questions about the paper? Anything else I can help you with the paper? We got about 5 minutes or so until our restriction enzymes are done digesting. I'm kind of mostly done with the lecturing portion today. Like I said, no more slides, but that means also the downtime. So we're going to get our gel electrophoresis started here in just a second. But the downtime while the gel is actually running itself is then going to take about 30 minutes. That's where you guys can do colony counts, collect your data from last week. It's also your chance to, if you have any questions about the paper or other remaining assignments for the semester, that's also your chance to kind of ask those, or even maybe if you wanted to, you could start working on a little bit of the paper or outlining some stuff and go through that with you as well. Questions about that? Okay. Then I would say start looking at page 78 of your lab manual, kind of just go through that, get ready to do the gel electrophoresis. I'm also probably going to stop the recording here, but I'm going to show you a few things real quick, just demonstrate some things. Loading a gel is super detailed pipetting work. You got to get your pipette tip into a little tiny well. And it actually takes a bit of technique to do that. You got to get your tip just above the well, but you can't go down into it or poke through the bottom. You can actually, if you put your pipette tip too far down to the well, you can poke through the bottom of it, and you put your sample in, and it just goes right out the bottom, and you lose your DNA, for instance. And it happens every year, by the way, for at least one well. So everybody's going to do some gel electrophoresis today, but also I have some practice gels up here for you to do some practice loading first, before you do the real actual loading of your sample. So over here at the sides, I'm going to have these practice gels set up in just a second here in these plastic trays like this. I'm going to put a practice gel in here. I'm going to have some practice loading dye with this as well. So, but you're going to practice with the practice dye. You're just going to load the practice gel. The other thing with the DNA is, so I'm going to set the practice dye out for that. But I have some real, what's called loading dye as well. I'm not going to set this out yet, so that you're using the right thing. But the loading dye is blue juice, in essence. You guys can see it's kind of purplish, actually. Helps you visualize your sample a little bit better. But it's also got glycerol in it, which is thicker and heavier than water. You're going to mix the glycerol with your DNA fragments. Then when you load them into the wells, it causes them to sink down to the bottom of the wells and stay in there. DNA is fully soluble in water, and you're just going to have this in basically water. So if you don't have a little bit of that loading dye in there, your DNA goes down to the bottom of the wells, goes into your well first, and then it starts to diffuse out and float away. In essence, you've got to use the load when these enzymes come out. You add the loading dye to it, according to the directions in your lab manual. Then after you've added the loading dye, you load it into the gel. But you got to add the loading dye for it to work effectively. Questions about that? All right. So read through that page 78, but then I'm going to set up the practice gels and we'll get started with that.